Okay, this video is going to review our 4 macromolecules that we can find in our body. So the first one's going to be carbohydrates, proteins, nucleic acids, and monomers. These are all polymers, which means they're large molecules, and they're built from smaller little pieces. And these smaller building blocks are called monomers. So when we're talking about a carbohydrate, what their building blocks or their monomers are, they're called monosaccharides. There's a couple of different examples in your PowerPoint and in your textbook that you should be aware of, such as glucose, fructose, galactose, the sugars from nucleic acids, so deoxyribose and ribose. Those are all monomers. We can link some of these monosaccharides together to get disaccharides. Di means 2, so we just have 2 monosaccharides linked together to get these. If we link more than 2 together, so 3 or more, we call them carbohydrates, or we call them polysaccharides. And again, poly means large and multiple numbers, so all of them are going to be polys. Proteins, what we're building, and you learned this from chapter 3 when we did transcription and translation. Proteins are built from building blocks called amino acids. There was 20 of them. I don't expect you to memorize them. They were given to you on a genetic code previously, but just know, and I spelled it wrong, just know that the building blocks are amino acids for proteins. We could call polymers of carbohydrates polysaccharides, like I said. We could call polymers of amino acids proteins, or we could call them polypeptides. So we can call these also polysaccharides. And we can also call proteins polypeptides. So when we were doing transcription or even replication, we worked a lot with the nucleic acids, so something like DNA and RNA, and their building blocks and what we linked together to get these nucleic acid strands were things called nucleotides. And we had 5 different options depending what type of nucleic acid we were working with. We could have had adenine, thymine or uracil, guanine, or cytosine. So those are our nucleotides, or building blocks of nucleic acids. And then again, the amino acids, these came from the genetic code, and there was 20 possible amino acids that we could link together to make our proteins or our polypeptides. There's 5 different monosaccharides. We can link those together, and I'm going to draw an arrow up here to make a disaccharide. And then even link additional monosaccharides to the disaccharide to make a polysaccharide. So if we have 2 or more, or 3 or more, we have a polysaccharide. If we have 2, we have a disaccharide. If we only have 1, we have a monosaccharide. The 4th type of macromolecule that we needed to talk about, or that we did talk about, were lipids. And lipids are unique because they do not have a true monomer. No true monomer. And if you go in your textbook or in the PowerPoint, you'll see exactly why. So we looked at a triglyceride today, which was basically, we'll start with lipids, a glycerol with 3 fatty acid tails. And then we looked at cholesterol, which is also considered a lipid. And it's a very rigid structure, and I'm not sure if this is its exact structure, but I know it's a 4-ring structure. So this would be cholesterol, and this would be a triglyceride. Both of these are lipids. We're not taking something that's very similar to each other, linking anything together to build a larger molecule. We just have a bunch of different types of lipids. And obviously I can't spell monomers. So no true monomers, because they look very different like I'm showing you here. And same thing with the phospholipid. If we had our phospholipid with the 2 fatty acid chains right there that we talked about, it looks different than cholesterol as well. And even our triglyceride. What else did I want to say about these? I think that's it. So that's just the basic differences. You need to go in and review some other components about what makes these things unique. So for example, if we talk about a carbohydrate, we said that they have to have a ratio of carbon to hydrogen to oxygen in a 1-to-1 ratio. 100% always that occurs. That's how you know you're looking at a carbohydrate. When we talked about an amino acid, they have to have 3 components. One called an amino group. That's why it's called an amino acid. Amino groups just contain nitrogen. It also has to contain a carboxylic group or some sort of acidic group, because again it's an amino acid, so we need the acid part now. So it has a COOH at the end, and then an amino group like NH2, and then we have something called an R group. And the R group for amino acids determines which of the 20 amino acids we have that we talked about. So those are the 3 components of every amino acid. R group is variable, and that's what's going to determine if it's leucine, glycine, valine, etc. Nucleic acids we touched a lot on in chapter 3. The couple rules that you need to know as far as nucleic acids is that we have certain bonding. So adenine always binds to or complementary base pairs with thymine. We learned this in chapter 3. And guanine always base pairs with cytosine if we're talking about DNA. When these base pair, I told you the DNA double helix is held together by hydrogen bonds. We're running 2 different strands. This is strand 1 of DNA. This is strand 2. So in order to get them to come together to form a double helix, they have to form hydrogen bonds. Adenine and thymine, they form 2 hydrogen bonds, and they're not true bonds, so I'm drawing them as dashed lines. And guanine and cytosine form 3 hydrogen bonds between each other. A couple other things to talk about is we can group these together, and whether or not they are what are called purines or pyrimidines. Purines are adenine and guanine, and pyrimidines are cytosine and thymine, or uracil if we're talking about that. Purines have a 2-ring structure. So smaller word, bigger structure, bigger word, smaller structure. So a single ring right here. So as long as you know the complementary base pairing rules and how many hydrogen bonds are forming, if we were to look at an example where we had two nucleotides base paired together, can we determine what specific nucleotide this is and what specific nucleotide this is based on this information up here? We certainly can. Okay, so first we look at the number of hydrogen bonds, and I see that there's 3 attaching these 2 nucleotides. Well, only guanine and cytosine form 3 bonds when they complementary base pair. So because we have 3 hydrogen bonds, we know that this has to be a guanine and a cytosine. Then we look up here and we see that purines is a guanine is a purine. That's a double ring structure. So without a doubt, we can say that this one is guanine and that this one over here is cytosine. Single ring structure. Let's do another example. So I'm going to draw a pyrimidine here we'll do 2 hydrogen bonds and a purine here. Okay, another thing I didn't mention, but as a rule, is a purine always base pairs with a pyrimidine. So you never have 2 purines together or 2 pyrimidines together. So if you looked at our base pairing rules, it's always a purine with a purine with a pyrimidine. Those are our only options. Okay, so I look here and I see we have 2 hydrogen bonds. So I know this has to be an adenine and a thymine if we're talking about DNA. Now we have to figure out which one of these nucleotides is adenine and which one's thymine. So if I know that adenine is a purine, purines are the larger structure, I can say this one is adenine without a doubt. And then I can look at this, it's a single ring structure, and it has to be thymine over here. And eventually you'll see they're basically all the same. So I could do another one because you only have 2 possible options. 1, 2. You're just going to see it repeated over and over again. Let's do 3 hydrogen bonds here. With the purine. Okay, so we see 3 hydrogen bonds. So we know it has to be guanine and cytosine because they form 3 hydrogen bonds when they base pair. Then we look and see which one of these is a purine with a double ring structure. And purines with a double ring structure has to be guanine. So this has to be guanine. The single ring structure over here is a pyrimidine and it has to be cytosine because G always base pairs with C. And we have 3 hydrogen bonds, so we know it has to be G and C together. So hopefully this helps you recognize a couple things. Let me see what else we talked about. Proteins and protein foldings. You can go in and read that in your textbook or slides. We did talk about lipids. Make sure you're aware of the difference between a saturated and an unsaturated fat. Again, carbon's unique in that it can form 4 bonds. So we say if it does form 4 bonds that it's saturated, it has the max number of bonds that it can form. If for some reason a carbon forms a double bond or even a triple bond with another atom, say another carbon, it's no longer saturated. It does not have 4 bonds. So unsaturated fats have double bonds, and because they have double bonds, it causes those fatty acid chains to kink. Because there's a double bond in the fatty acid chain and it kinks, they can't stack nice and neatly as molecules. So if we think about an unsaturated fat, it's going to be liquid at room temperature, all because of that double bond. Saturated fats have nice and neat fatty acid chains, and they can stack really nicely and neatly on top of each other. So those would be solids at room temperature, something like Crisco or butter. But again, not really a true monomer. Also, you need to be aware of the different types of things that can be considered a lipid, like cholesterol and different steroids. So go back through and read. These are some of the harder concepts. Reach out if you have any other questions regarding our macromolecules.